It's an honor to be here. It has been a great pleasure, and I've learned a lot, and I hope to learn a lot more in the future, in future discussions. So as he just said, I don't know, I wanted to introduce what I'm talking about, but he just said it. So the work I'm going to talk about is actually about the postdoc work that I did with Jose Manuel Alonso in New York. And it's about, as the title says, about cortical topography in the visual cortex. So when you look at this image that you might be familiar with when you live in Seattle, there are cells in your brain that selectively respond to edges at different orientations and spatial locations. And we know that, as you will know also, because several decades ago, Hubel and Wiese made the fundamental discovery that cells in your primary visual cortex, so here at the back of your head, are orientation selective. This is shown here at the right, where you see a figure from the original work from Hubel and Wiese, where you see on the left the different edges of the visual stimulus having different orientations, and on the right you see the neural response of this particular cell. And this particular cell had a preferred orientation of a vertical bar. And this response feature in cortex is quite remarkable for several reasons. One is that it's at least in higher mammals, it's an emergent property of cortex as brain structures upstream to the visual cortex, so neurons in the LGN or in the retina, are in general not orientation selective. So it really emerges in cortex. And secondly, orientation selectivity has been found in all mammals where it was studied, so in mice and in rats and basically all mammals. So it shows that it's a fundamental aspect of visual processing. And despite the fact that we have all the cells in V1 are orientation selective, the topographic organization of the preferred orientation, so the orientation that the neuron prefers, is not universal. It's actually species dependent, and it comes in two flavors. Roughly two flavors. So in some species, such as cat, monkey, and us humans, we find so-called orientation maps in V1. And in these maps, the preferred orientation, which is shown here as a color code, is systematically mapped onto the cortical surface in a so-called pinwheel fashion. And this is shown here on the left, where you see a top view onto cat visual cortex, and the color encodes the preferred orientation of a given cortical region. For example, all the blue regions would prefer a vertical bar. And these maps are usually were measured with optical imaging, so intrinsic signals. As I said, it's a pinwheel pattern where at the blades of the pinwheel, the orientation changes smoothly, and then you have a discontinuity at the center. And this kind of rich structure is in quite contrast to other mammals, such as the mouse that you use quite a lot here in the Allen Institute, where you have a so-called salt and pepper or random organization of orientation preference. And remarkably, despite the fact that orientation selectivity was discovered decades ago and orientation maps were discovered decades ago, the underlying principle of why we have these maps, or in general, how cortex is organized topographically, was still slightly elusive, in particular in the question of the orientation map. And this is what this talk is about. I think we now finally understand much better how these maps are organized, at least in these species which have orientation maps. So this brings me to the specific questions. As already in the introduction, we have these very fundamental pathways in the system, so the on and off pathways, and they are quite linked to the orientation preference in V1. So what are the on and off pathways? They originate in the retina. So after the very first synapse, you have cells in the on pathway that have these concentric receptive fields. They have an on center. They are selective for light increments. They are not orientation selective. Then cells for the off pathway, they're kind of the mirror image of the on pathway. And to some extent, as that they are selective for light decrements, they have these concentric receptive fields. They are not orientation selective. And then these cells project to the LGN, where nothing really much changes in this respect. And it's really from the LGN to cortex where the fundamental transformation takes place. And what takes place is the following, that the LGN afferents arrive in layer 4 mainly. And in this layer, you find the so-called simple cells, which have these elongated off and on receptor subfields. And these cells are orientation selective for the first time. And what links the on and off channel or on and off visual responses in cortex to the orientation selectivity is the fact that the arrangement of the on and off subfields kind of predicts the orientation preference. So this cartoon cell here would predict, or would have a preferred orientation, like of a vertical bar, would prefer a vertical bar. So on a single cell level, there's a strong evidence that the on and off visual responses in cortex are linked to the orientation preference. However, what was not known is whether on the population level, like across the cortical surface, whether there's any particular architecture for the on and off visual responses. And this was the particular question that we wanted to address in this study. So we asked ourselves, the question is the cortical organization of on and off central for the topographic organization of V1? Or in the first place, is there a cortical organization for on and off? And what do I mean with this? This is shown here in this little cartoon, where you see the different cortical layers in V1, and you see the on and the off algae and afferents arriving, and the middle layers where you then have the simple cells. And the question really was whether across the horizontal dimension of layer 4, is there any particular organization for on and off? And this question was around in the field for a little bit, but it wasn't really answered because the method that were used to study cortical maps, so this optical imaging and so on, they could not really read this signal. So they usually, these optical methods, they only read the activity of the upper cortical layers. So one couldn't really use that method to address this question. So therefore, we decided, okay, we cannot use optical method, but let's use a different method where we used electrode probes, so linear electrode probes. And in the first experiment, we want to understand the organization of on-off in a cortical column. So you probably all remember that when you record perpendicular to the cortical surface, that all cells will have the same orientation tuning, or very similar orientation tuning. So it's the definition of a cortical column. And our question was, like, if we record now along this column, how within the middle layers is the on and off retinotopy organized? To do that, we used the CAT as a model system. So we inserted a 16 electrode probe perpendicular to the cortical surface in vivo, and then we mapped receptive fields and so on. And despite the fact that this has only 16 channels, not these 300 that you now have, or thousands with your pixel probe, we could understand something of in V1. Because when we looked at the receptive fields, they were mapped with white noise, and this is, so these are 4 receptive fields measured within the middle layers of CAD V1. And so this is retinotopic, like visual space, and then the blue subfields are the off subfields, and the red subfields are the on subfields. And these are like simultaneously recorded of 4 electrodes. You see that there's a diversity in the receptive field property, which is good for neural coding, but that the retinotopy, so that the position in visual space of the red and the blue blob is kind of remains rather constant throughout the depth of V1. And this is shown here in the average, where you see that the blue and the red kind of are the center of this little image here. This was very remarkable, as it shows that there's a columnar organization for on and off in V1. So there's a clear structure in the vertical dimension of V1. So it was not just random, as previously assumed. And this was very important because now this opened up the possibility that there's also a horizontal architecture of the on and off visual responses, and that could be very important for the cortical topography in V1. And to address this, we used again multi-electrode arrays, and now instead of recording perpendicular to the cortical surface, we made tangential recordings. So the cat brain is very shallow, so we entered with a very shallow angle and recorded so the aim was to record as much as we could within the middle layers in the horizontal dimension. And just to give you a little feeling for the spatial scale of our question and the probes. So we had a 32-channel probe, which the electrodes were separated by 100 microns, which gives them, they were 3.1 millimeter long. So, they're just the recording part. Of course, the electrode was much longer. We inserted them from the posterior to anterior side in CAD V1, and this right image shows you kind of the recording length, or like the dimension that we could record. So we could really, using this probe, record a long stretch of the cortical organization in V1. So we could record different pinwheels and so on and so forth. but at a higher spatial resolution because we had an electrode every 100 micrometers. And so what did we measure? We measured very basic things. We measured orientation tuning using 16 bars, like moving bars in 16 different directions. And then we measured the tuning curve of that. So this is one of these neural responses. We see this is time. These are the different directions. And this cell preferred, or like was strongly responding to a horizontal bar moving downward. This is the polar representation of this tuning curve. And from this, we could extract the preferred orientation. And then we also mapped the on and off visual responses using light and dark sparse noise. So we had to map on, as I just mentioned, the on responses are the responses to light increments. So to test this, we had like a dark screen with small white spots on it to get the on response. And to get the off response, we had a white screen with the dark squares, and then this gave us the off receptive field. So from these on and off receptive fields, we calculated the on minus off receptive fields to get kind of the individual subfields. And then we calculated from this using 2D Fourier transform a preferred orientation, which would be also roughly this angle. And we compared this to the orientation map. So this is how we linked the cortical organization of on and off to the orientation map. And now I want to show you one example of our recording. And if you want to remember anything from my talk, then this should be the talk. And the title already tells you the solution. So we discovered that the on and off retinotopy, like systematic changes across the cortical surface, kind of explains the orientation preference map. And I will walk you through this figure. So again, we have a bit more than 1 millimeter of cortex in the horizontal dimension here. So these are the recording positions. These are the off receptive fields that were measured simultaneously at all these positions. They look, the retinotopy changes smoothly a little bit because there's a retinotopic map. When we looked at the on responses, things looked a bit different. So now the on receptive fields looked a bit more jittery. But okay, this was very interesting, as you will see later. But remarkably, when we calculate the on minus off receptive fields across all these channels, then you could see something that resembles an orientation preference map. For example, this cell here would prefer a horizontal bar, and then they would kind of change it, and this would prefer an oblique bar, and then over here, this would prefer a vertical bar, and then it would go back to a horizontal bar. And this is very interesting because the spatial scale of this kind of resembled actually the spatial scale of the orientation map. And when we then compared this to the actually orientation map that we measured with moving bars, then we discovered that it was actually a perfect match. I mean, in this particular example. For example, here these are the polar representation of the tuning curves, and you see that this cortical side preferred a horizontal bar moving downwards, and so on, and then it shifted around, and then it came back to the horizontal bar. And these magenta lines as circles are the prediction from the on and off receptive fields. So this really showed that one thing, there's a systematic mapping of the on and off retinotopy in cortex across the horizontal dimension, and that this interaction of the on and off retinotopy kind of predicts the orientation preference map, so links to the orientation preference map. In addition, so this was actually the aim that the experiment that we aimed for. So we're happy at this stage, but then there was more to it, and this is what was more to it is shown here at the top, where you see that off and on they looked a bit different, as I mentioned just briefly. So off seemed to be a bit more stable in retinotopic space than on. And it turns out that this is actually quite important. And I show you this in a different representation where I have a little animation that goes through kind of some part of this data. And what you see here, or should see, is that the off, although across channels, so like one of these animation frames is one of these recording positions, and you see the millimeters, that off kind of, although it moves slightly because the retinotopy is changing, but that on kind of rotates around off, and that due to this rotation, you get this change in orientation preference. So kind of, we call it at that time like this off is the anchor of the retinotopic map because it moved slower. And we reported this a few years ago at SFN, but at that time we didn't really appreciate the importance of it. So we said, okay, maybe it's a cat-specific thing because these recordings were from the cat. But it turned out that it's not just a cat-specific thing, but it seems to be a fundamental or a principle of how V1 is organized in species with at least orientation maps as we know so far. Because subsequently, it was found in several species. And this is what is shown here on this slide. So this is our cat data, which this is a different representation of the same data. So where you kind of sum together 10 of these receptive fields, and you see that off is kind of centering and on is moving around. These circles here are the peaks of the on and off subfields, so the centers of the on and off subfields. And you see that the off one is kind of in the center. Again, an on moves around. This was cat. We subsequently tested it in monkey, and we found basically the same thing. And more remarkably, that at the same time when we conducted the study, there was independently the lab of David Fitzpatrick and the Max Planck in Florida conducted a beautiful study using a very different approach in tissue. So instead of using multi-electrode arrays, they use calcium imaging. But so these tissues also have orientation maps. And basically they found exactly the same principles. And one of these plots is shown here where you see off, it's in the center, and on moves around. And also in their study, they find that the relation of on and off across the cortical surface is kind of explains the orientation map, both in the horizontal dimension as well as in the columnar architecture. So this gave us a real, this was our data, and it was really nice to see that a different lab using a completely different species and analysis method kind of found the same thing in terms of reproducibility. So we were kind of sure that this is really true. Okay, let me summarize the first part. I hope that I convinced you that the way on and off is organized in cortex, that this is fundamental for the orientation preference organization. This little cartoon shows this. So these are supposed to be 3 recording sites, and the orientation change from one to the next is due to the way in this particular case, the on moves around, and then off is more stable. And currently, the hypothesis is that that this could be actually very simply explained by the way the thalamus projects onto cortex. So it's not just a random projection from thalamus to cortex, but there's some structure in it, such that, for example, some LGN afferents here with this retinotopic position would spread to like a region in cortex, and then the on would be supplied from a different sets of pools in LGN. So this is very nice, but the orientation preference map is not the only cortical map that we have in V1, but there are actually more maps, and we were then interested in how the cortical organization for on and off relates to other cortical maps. And a very important one is the ocular dominance map. This is shown here on the right. So here you see again the orientation preference map. This is the ocular dominance map, and what the ocular dominance maps shows is that, what it reflects is that, for example, in this black region, there would be cells or neurons would respond predominantly to the contralateral input, whereas in the right region, it would be ipsilaterally dominated. And these two images were actually done, or cortical maps were recorded in the same cat. So this is also cat. It was actually the same experiment. And in this study, they compared the relationship between these maps. And it turned out that they're actually strongly related. And this plot shows this this plot shows this by representing these two maps superimposed to each other. So these colorful lines are now the contour lines of the orientation preference map. And the gray shaded region here is the contralateral dominated region. The black line is the ocular dominance border, and the right region is the ipsilateral dominated region. In this kind of blown up version, the relationship is not so evident, but when you zoom in, you see that these maps are orthogonal to each other. So, for example, if this is the contralateral dominated region, ipsilateral, so from over here to here, so this is the border, the ocular dominance map changes drastically, but the orientation preference map, which are the color lines, changes hardly. So when the orientation map doesn't change, the ocular dominance map changes, and vice versa. This suggests that, it shows a strong relation, but it also suggests that whatever mechanism gives rise to the orientation preference map will probably also be very related to the way the ocular dominance organization is organized. This is what we then looked at in our data. And basically, we pooled our data into two configurations, or two. So we just poked into the in CAD V1, and the cortical organization is not a crystal, so we didn't really know where we ended up every time we recorded there. But it turned out that we had recordings that were either parallel to the ocular dominance columns, so where our recording electrode was along a border or almost parallel to a border, or that we had recordings that where we crossed the ocular dominance border, so where we crossed multiple borders. And it turned out that this, these different recording configuration was actually quite important and revealed us another architecture in V1. And I'll show you first the recording from the parallel examples. And this is again a recording of roughly 2.5 millimeter of cortex in the horizontal dimension using the technique that I just mentioned. And on the bottom are the off receptor fields of this particular recording, and above are the on receptor fields of this particular recording. And what you should see here, or should note, is that on and off responses are not strong at every cortical position. So they form kind of domains. For example, this would be an on domain that has a certain size, like roughly 0.5 millimeter or so, where on is kind of dominating the responses in cortex. Likewise, you have off domains where off is strong, particularly at this point of the recording. So there are on and off domains in cortex. And as it turns out, when you record parallel to the ocular dominance border, that these domains are anti-correlated. And this is shown here in these colorful lines, in the red and blue line, which kind of just represent the maximum of the receptor field. And you can see that when you go across the cortical surface, that off is strong at this position, and on is weak at this position, and so on and so forth. So these two lines alternate, showing that these on and off domains are anti-correlated. And this is important because the picture completely changed when we looked at the recording perpendicular to the cortical surface, to the ocular dominance columns. So now the picture is a bit more complicated because now we have two eyes. So these are the on and off responses from the IPSI eye. These are the on and off responses from the contra eye. And maybe don't focus on so much on this one because it might be a bit confusing. But what is important is that now, instead of being anti-correlated, on and off were correlated within the same eye. And this is, for example, if you just focus here on the solid lines, they peak roughly at the same cortical position here in this, and then they go down because now this eye is not dominated anymore. Then they go up because this eye is dominating. They peak roughly at the same position, and then they go down and they peak roughly at the same position. And likewise for the IPSI lateral eye dominated region. So it looked as if, depending on whether you cross a border or you're going along, that these domains are differently organized. And we quantified this in many details, the widths of the domains and so on and so forth. But it basically boils down to what we concluded from our data, that these on and off cortical domains, that they run orthogonal to the ocular dominance columns. And this is shown here in this little cartoon, where you have on the left the left eye dominated column, and here the right eye dominated column. This would be the border, and then you have the off and the on domains that run orthogonal to that. And this architecture was kind of, when we looked at it, it was quite interesting, but it suddenly also could explain us why, as I mentioned, why the orientation doesn't change across the border. So why is that the case? And it leads us to another kind of insight. So what the cortex, or what V1 is doing at this border, is that it actually matches the on and off polarity between the two eyes. So the border, you have binocular cells, and most of these cells are matched in terms of retinotopy, and this is shown here on the right example, where you see 4 consecutive channels in the binocular region, and you see the on and the off responses, and the color map is the ipsilateral eye receptor field, and the contour map, the contralateral eye input map, and you see that they are well overlapped. So as if the cortex kind of what it tries to do is to have a continuous representation of the on and off retinotopy at the border, and by doing this match, you have a hard time changing the orientation preference because the on and off organization is kind of defines the orientation preference. All right. With this, I would like to summarize what I presented you as our new insights into the cortical topography in CAD V1 or in species with cortical maps. I showed you that there's a columnar organization in layer 4, so that on and off are not just randomly organized, but they kind of form columnar organization. I showed you this from CAD, but I mentioned that also it's true in tree issue, as David Fitzpatrick has shown. So very likely it's also in your brain. I reported that we have these all cortical domains for on and off, and that they run orthogonal to the ocular dominance organization. And most important, that changes in on and off retinotopy across the cortical surface give rise to the orientation map. So finally we understand that. And that remarkably, we still don't really fully understand what this off being the anchor of this whole game is. We don't really, I mean, it's current research what that means. It could relate to the fact that the off channel has a higher spatial resolution, and it could well be that it uses the off as the anchoring of the retinotopic map because it has a higher spatial resolution. But this is just my best guess right now. And in general, I would like to conclude my talk with a statement that currently we're working on. And I mean, our current belief is, or the hypothesis is that this cortical organization that we discovered in the middle layers could easily be explained from projections from the thalamus, and that in general, that it's probably very important how the different pathways that are in the thalamus, how they project onto the cortical surface. That this is probably very important for the topographic organization of cortex. In our particular case, the on and off channels in the visual system. With this, I would like to finish. I'd like to thank you for your attention and acknowledge the people that really made this possible. So first of all, I would really like to thank Jose Manuel Alonso. He was and is a great mentor, and all this work was done in his lab. It was a fantastic time, probably a postdoc period like everybody dreams for. It was inspiring. Then I would like to also thank Jin, who taught me everything about these CAD in vivo experiments, because as I was introduced before, I did my PhD in computational neuroscience, and before joining his lab, I knew a little bit about in vivo recordings, but definitely not about CAD. So Jin taught me everything about it and helped with the experiments and the data analysis, and also Yushi, who helped a lot with the experiments. And finally, I would like to thank, of course, the funding agencies, the State University of New York, the NIH, and also this is the German Science Foundation, the DFG, which supported me for a while, not only actually me, but me and my family, and who made this fantastic experience happen. Thank you.
So great talk, thank you very much. So you showed us these results for the layer 4.
Mm-hmm.
I'm wondering if you have any data or know of any data or could speculate how would this look like in the layer 2-3 or other layers?
Yeah, so for the horizontal, I mean, usually the classic idea is that in the upper layers you have the complex cells where on and off are not segregated anymore. So it's kind of a mixing there. I remember that when we did these perpendicular recordings, so the columnar organization, that we did find that also in the upper layers it was kind of not just, there was a bias. So I don't know, if there was an off in the center in the middle layers, that it kind of remained also in the upper layers. But in general, these upper, the neurons in layer 2-3, they are more complex, so they in their receptive fields on and off are more overlapped. So it's kind of washed out at this stage. That's why it was also so difficult to measure with optical imaging, because to really see this segregation of on and off, you had to go to the middle layers. So therefore, to answer your question, I think in the upper layers it's going to be slightly different. probably the on and off domain is going to be still reflected there, but since these are complex cells that merge on and off in the same retinotopic space, probably not as clean as in the middle layers.
So this is something of a speculative question, I think. So based on the organization that you've described, which is really beautiful, do you think of it more in terms of serving a particular computational role on the one hand versus being maybe a developmental expediency or even something that was just locked in through evolution early on and we have to build around it?
Yep.
So what are your thoughts there?
Very good question. Yes, I don't know the answer, but and we're working on that. But it's, I don't know. I would hope that it has some functional role. I mean, it's so beautiful. I mean, it's hard to believe that this is just there by evolution. As I mentioned, that OFF is the anchor of the retinotopy could be related to the fact that OFF has this higher spatial resolution. And that kind of makes sense if you want to map the retinotopy, like the visual world, as precise as possible to cortex. This would make sense. Okay? But why you have orientation maps? I mean, I really don't know. So the OFF anchor kind of makes sense in the sense that it has a high resolution. But why do you have to arrange orientations in such a beautiful arrangement? I really don't know. But it would be great to compare this to the mouse and see.
Do you, all of the data you showed, I gathered, was from responses of cortical simple cells in layer 4, not from the thalamocortical afferents directly themselves.
No. Yes.
Do you have any view about whether the topography of these anchoring off responses is continuous or not? What you described over a long distance in some of the electrode penetrations, the off responses hardly moved at all.
Well,
And if you were to continue that, would they move continuously or would they move in jumps?
Yeah. I mean, we have many recordings and the off responses do move. In this particular example, they were rather not moving, but they do move because there's the overall retinotopic map. So they do follow that. And in some recordings, depending on where we penetrate, retinotopy moved faster than in other recordings. So we do have recordings where retinotopy kind of is continuous. So I would, we have only like this 3 millimeter of cortex, but I would say it looked rather continuous. So we had, I didn't see like sun jumps. Maybe at the border of A17, like where you change from V1 from A17 to 18, where retinotopy goes in the other way, there was maybe a jump. But staying in the same cortical area, we just saw a continuous mapping.
Is the higher resolution for the OFF something which can be explained from the statistics of natural scenes?
The higher resolution, I'm not so sure, but so OFF is in addition is dominated, so it's stronger in V1, and this can be explained by the statistics of natural scenes, so that OFF is dominating natural images, and OFF contrast is more there. But the higher resolution, I mean, this is work that we published in the PNAS paper. We discovered a nonlinearity in the on pathway that kind of blurs the on representation. And our take at that point, or still is, that due to this nonlinearity, you kind of blur the on pathway. How this relates to the statistics of the natural scene, I think is still ongoing work.
I can't resist asking, what do you look for next as you go to higher thalamus? Would you expect this sort of on-off structure to repeat or something utterly different to emerge?
So in the cat, I mean, that's a good question. I don't really know whether it should repeat or not. Maybe yes. I mean, if the fundamental role is to kind of have a precise mapping of the retinotopy, then yes, I would assume that why not use that feature again and again? But it's hard to speculate. But I think what would be really interesting to study, and I think what you could easily do here at the Allen, is to see whether some of these principles actually hold true in the mouse. Maybe not. I mean, you don't have maps, that's for sure. But whether off is the anchor, but this is, maybe it's also implemented in the mouse visual cortex, just to a different degree. Maybe that's, a defining difference between these two species. That would be really interesting to study, and I think you have all the machinery to do that in a very quick time.
On that note, let's thank Jens again.